discuss the story of insulin, right? Insulin is a hormone secreted from pancreas. In the previous class we have discussed. And what is the story behind it, right? You can see this here from the beginning. In the year 1868, the Paul Langerhans is the person who studied on the functioning of pancreas. He is the professor of pathology at Freiburg University. At Freiburg University is the pathology professor working in the Freiburg University. He had chosen a specific topic that working on the structure of pancreas, and how the structure of the pancreas and how it is working. Later, he noticed the special kind of cells in the pancreas, right? And these cells are not like the other cells in the tissues, and these cells are richly supplied with the blood, and these cells are functioning differently, and these cells are freely float on the blood itself. These are the special qualities that attracted the particular person, Paul Langerhans, to study on the particular gland itself, right? And later he noticed that these cells he named as a islets of Langerhans. His name is the Paul Langerhans, right? On the name of his scientist, he called those cells, particular cells which are different from the other cells in the tissues, which are freely float on the blood and richly supplied with the blood, and those are, he called it as a islets of Langerhans. Why he called as the islets of Langerhans? The sense of islets means island. Island means we are seeing that generally around a particular piece of land which is all sides surrounded by water. It is generally called as island. So these cells also, like, they freely float on the blood, and around the cells there is a blood itself. So he compared the cells in the blood and land in the ocean itself. So he called it as a islands of Langerhans on the name of his scientist. Later, the few scientists, they have eagerly waited the functioning of the pancreas, and so that the pancreas was removed from the experimental animal, the dog. They have chosen a dog as an experimental animal to functioning of the pancreas. So he has removed the pancreas from the experimental animal. What happens automatically when the pancreas has removed from the experimental animal dog? They develops the diabetes. Diabetes in the sense of normal blood sugar. Increase the level of glucose in the blood more than the normal. That is generally called as diabetes. Generally called as blood sugar. So whenever they remove the pancreas from the experimental animal, in that animal blood sugar levels are highly increased. That they were noticed. Later, the another way, they tied up the pancreatic duct. Pancreas is the gland which is present below the stomach and attached to the duodenum of the small intestine. When you observe the pancreas has a duct that is opens into the duodenum. What they have done, they tied the duct which is closed to the duodenum, and what they have done, they cause the pancreas to degenerate. Whenever the duct which is arising from the pancreas had been tied, that causes the pancreatic gland should be reduced in size, degenerate the pancreas size. But islets of Langerhans remains normal. Islets of Langerhans remains normal, even though they tied the pancreatic duct which is connected to the duodenum. The size of the pancreas reduces, but still the islets of Langerhans, their functioning is very normal, right? So that the animal also would not develop no diabetes. Right? Even though the duct arising from the pancreas had been tied, only they see the size of the pancreas degenerated, but in the animal, no diabetes would develop, right? So finally, what they have done in 1912, the islets producing secretions are released into blood. They finalized that. By this activity, in the 1912, they finalized the pancreas ducts, even though they have tied, still the islets function is normal. So they come to the conclusion that the islets of Langerhans or not have the contact with the duct. Their secretions directly release into the blood, right? So that was finalized in the 1912, right? In the Latin, insula means island. Insula means island. So that particular secretions from the pancreas that they named as a insulin, right? The secretions from the particular cells called insulin, because insula means island. Later, after 10 years, Toronto, Banting, Best and MacLeod, these are the four scientists, they have done the experiment togetherly, right? In the 1912, they have done the conclusion. After 10 years, in the sense of almost in the 1922, Toronto, Banting, Best and MacLeod, they have togetherly extracted the insulin. Finally, they succeeded in the extracting insulin from the experimental animal whose pancreas duct tied. Right? What the experimental animal previously they have taken from the same animal, the whose duct has tied, they have extracted the insulin, extracted the secretions from the islets of Langerhans, and they given those extractions, they are given intravenously to the same dog. What the chemical substances are extracted from the pancreas, the same chemical substances are injected intravenously to the same dog, and whose pancreas was removed. Then what happens? The substance we kept it alive, and a dog does not having the diabetes, and the blood sugar in the blood level, glucose level in the blood will be low. That's what they identified. Low level of blood sugar. Right? This is what finally they can come to know, that the particular cells present in the pancreas, they are secreting the insulin not directly from the duct which is arising from the pancreas. They are releasing from the directly from the gland to the blood itself. So that even though they tied the duct, the pancreas size only decreased, but still the islets function is very normal, and the experimental animal also not developing any diabetes or the blood sugar, right? And finally, they concluded that these are the particular cells present in the pancreas. They secreting their secretions directly into the blood, which are controlling the level of glucose in the blood. And nowadays, the pancreas functions, and the people, those who are suffering from the diabetes or the blood sugars, usually they are taking the intravenous injections in the morning after breakfast or after lunch and dinner itself. They will be taken a sufficient quantity of insulin to reduce the level of glucose in the blood. But nowadays the technology has developed that the insulin secreting bacteria also be available, so that bacteria can be injected into the body, that can be stimulating the increasing of the insulin, and it can automatically controlling the level of glucose in the blood when the person is consuming any kind of food materials. This is regarding the story of insulin.